Good. I'm in the middle of going through your guys' exams. HBY. What's HBY? I know a lot of them. I'm so lingo, right? I'm so lingo savvy. What's HBY? Happy birthday. Oh, how about you? Oh, I'm fine. Thank you. All right, cool. Learn another one. Every day I learn something. So I'm working through your exams. I'm about halfway. I'll have those done today. I don't have anything on my docket right now that's supposed to be standing in the way of that. I usually return them a little faster than this, usually within a couple days, but I just got stuck doing a bunch of things that didn't give me the time to sit down. I do make comments on your exams in various lengths, depending on, what I observe in that first exam. Like, for example, if you didn't do well, and I noticed, because it times you, you only spent 20 minutes in the exam, I might suggest something like slowing down, or maybe your essay answer was good, but there were a lot of mistakes. I might ask you to make sure you proofread, just little sort of helpful comments. But anything you don't understand, you'll be able to get into them when I'm done today. You'll be able to look at them, and you'll be able to look at the comments too. So sometimes on the rubrics, I didn't even go in and, if you scored perfect on the essay question, I didn't go in and click all the things on the rubrics, but I made the comments if I had any down below. So if you have any questions about the exam, about the answers will be shown as well. It'll be your answer, whether it's correct or not, and then, of course, the actual correct answer, or anything about the essay or the calculations. If you did not do the Hardy-Weinberg, or if you did not get that one correct, especially if you didn't get the calculations correct, because there were a few of you that did, you wrote out the essay part of it, a couple of points, or what was it, 4 points that you get for telling me about the stipulations and after 15 generations, but you did the calculations wrong. Make sure you get with me and we go over that again, all right? Because there's a very good chance that that's going to be on the first lab practical. I find that an important part, because that's a quantitative way of demonstrating evolution. So through natural selection, right? Well, at least evolution. It's up to the scientists to demonstrate it's through natural selection, but I do find that important. So please get with me on that, okay? Make sure we go over those again. In the exam, I show you how to work out the problem, but it's kind of in a shorthand. So if you made a mistake and you don't understand why it's a mistake or why the correct answer is the way it is, please, we can do it online or however you want to do it, okay? All right, so back into the protozoans. Anybody got any questions or anything like that before we go? Before we go into the lecture, we're not going. Nope, okay. So again, take a look at those exams, or be looking for those later on today. So again, the picture here is so much better than it is in class. I know that projector, I've been telling them for years that there's something wrong with the lamp, but look at the layering is what I mean. Is my pen working this morning? It is. Look at the layering that occurs. Now, if you were somebody that could examine the rate at which these foraminiferans, remember, these White Cliffs of Dover, what the White Cliffs of Dover are made out of is, I didn't write it right, chalk, right? I didn't write it on here. Calcium carbonate, the stuff that used to be in the classrooms, which I actually prefer over the markers for some strange reason. But if you could figure out how fast it is to create, say, a meter, so 1 meter of depth, you could kind of figure out this geological time scale in some degree, right? You could say, I have no idea what it would be, but you would say that it's certainly been longer than a month since that cliff has raised that far up. Now, yes, the sea level could have gone down, and it has gone down. During the ice ages, it's gone down. It's actually up now because of the fact that we're not in an ice age. So nonetheless, the reason, if you could show that the last ice age or the last low seawater level was X amount of years ago and correlated to how long it takes to make these cliffs, you could demonstrate that the continents are moving, number one, but they're moving pretty slowly, aren't they? That's what's all around you in the world, folks, when you think about science. It's all around these cool little clues that Mother Nature just happened to randomly lay down for us. And because we developed that bigger part of our of our brain, we can look at stuff and ask questions. And I think that's what's always fascinating me about science. So it's enough of me on the soapbox for today. But everybody remembers the foraminiferans. They were all, it looked like a broken bunch. Like some of them were more or less intact. Now, we didn't see the pseudopodia, the little pseudopodia that stick out between. Remember, you can have blunt pseudopodia like amoebas, or you can have thin ones, because those were, when they make that slides, those are destroyed. But we did see the calcium. We see these little flecks of calcium carbonate tests or shells, don't we, right? That's pretty cool. Years ago when I was teaching this class, I would hold up a piece of chalk and I would say, so these ancient organisms, they gave their homes for you so that you could learn about them. And nobody would laugh. All right, now the last member of this group, where are we? We're in the Sarclades still? Yeah, the last member of this group, or the Rhizarians, is going to be the Cercozoans. And this is the first book that's ever put the Cercozoans in them. I had heard about Cercozoans, but wasn't really, shall we say, impressed about them until we started using this textbook, and then I kind of put two and two together. I said, oh, that is kind of cool. So anyway, let's take a look at them. Morphologically, they're kind of diverse. They don't look like they're particularly exciting looking. They have a flagella. You can see that little flagella out there sticking out to the right. That's a nice picture, actually. And then, or pseudopods. So they either have a flagella, or some of the species, not this one here, obviously, but some of the species move with those, blobs or extensions moving out. We would say that their distribution, soil, marine water, fresh water, we would say that their distribution, and I hope I spell this because I'm trying to sound so fancy correctly, but ubiquitous, I believe, is the word. So when you see that word, if someone says something's ubiquitous, it's all over. Or the other word for it, many times I see, is cosmopolitan. This organism has now that you're looking, cosmopolitan, there we go. Now that you're looking up information about your field notebook, that's what those words mean. Ubiquitous looks more like a Mexican food, so that's probably not spelled right. Probably an I not supposed to be in there, but anyway, you'll find out. So common in soil, marine, freshwater, that's cosmopolitan, or it's ubiquitous. Flagella or pseudopods. Endosymbiosis. You remember that whole engulfing thing that we talked about, where cells, eukaryotic cells engulfed a photosynthetic cell, and some of them engulfed both that and an aerobic cell, which that became the mitochondria. This was actually introduced to you, I'm sure, I hope, back in Bio 1. I know I talk about it a little bit in Bio 1, but that whole idea that a symbiotic relationship that's specific because it's something gets engulfed, okay? And when scientists come out and speak about those sorts of theories, there's evidence to support that. Although there are theoretical scientists, mostly in that physics field, but they have enough knowledge that they can make reasonable theoretical predictions. That's the whole idea behind inductive and deductive reasoning. But what's interesting about Cercozoans, specifically this one, I love the name too, Pollinella, right? Chromatophora. Now, the Pollinella sounds to me like somebody in this field named it after their daughter, Pauline, or something like that. Who knows? But anyway, the chromatophora, chromato is color, and phora is like a part of that word morphology that we talked about, which is what something looks like. But phora specifically is to have that. So Paulina chromatophora has something colorful. Right? Isn't that kind of cool how they come up with these scientific names? That's how you can remember them sometimes. And in the case of Paulina chromatophora, what it has is a chromatophore. And notice that the chromatophore is colored green in this particular case. So the chromato in the chromatophore in this case is green. It doesn't have to be green, but oftentimes it is. So if you were to make a reasonable prediction using that inductive reasoning based about what you know about Bio 1 and the things we've talked about, how does Paulina chromatophora make its living? What does it do? What nutritional mode do you think it uses? Yep. See, it's that detective stuff, right? Because that's probably chlorophyll, right? You would go on and detect that, or you would go on and test that. Now, the other piece of the data here that I want you to look at and see if you can't figure out what it's telling you, it's in the parentheses. It's surrounded by a membrane. Okay, big deal. Lots of things have membranes. We've talked about that. But this membrane is a peptidoglycan membrane. So Paulina chromatophora is a eukaryote. Okay, it's a eukaryote. It has a nucleus, just like we've been in the eukaryotes this whole chapter when we're talking about protozoans. But how is this evidence of endosymbiosis, number one? And number two, what kind of critter was endosymbiosisized? What kind of critter was engulfed? I should put this on the exam. That's a good question. Remember that. There's two things to that, right? So how does this demonstrate, how come we can say, oh, cercozoans are, evidence of endosymbiosis? It was only a chapter ago. Madeline, are you? Did you just turn it on and go back to sleep? You're just being polite, or are you awake this morning for me? I always love your honest answers. I have no idea. Well, do eukaryotic cells have peptidoglycan membranes? I mean, we have membranes, but we have that weird peptidoglycan stuff. You remember, that's the stuff that shrinks. Yeah, we don't have those, do we? Us eukaryotes don't have those. So number one, if Paulina chromatophora is a eukaryote, but it has inside of it a peptidoglycan membrane, that means it probably must have engulfed something, right? It doesn't mean it's certain, right? But it is a pretty good evidence. And what kind of critter do you think it engulfed? Did it engulf, for example, another eukaryote? And if not, why not? I'm trying to use my computer as a side lighting, but it keeps turning off. That's why the light changed like that. I mean, eukaryotes don't have a peptidoglycan membrane, do they? Are there any? No, I don't think there were any, right? No, there's none. But who does? Well, that's a good start because, of course, it does photosynthesize. But remember, plants are eukaryotes, so their membranes, like ours, they wouldn't have peptidoglycan. But there is one group of organisms out there that we do find peptidoglycan in their membrane. Remember, again, when you stain it, that Gram stain that we talked about, that's the thing that shrinks a lot because there is a lot, or there's just a little bit, so it just shrinks a little bit. So, yeah, it engulfed a photosynthetic thing, but there are things outside of plants that are photosynthetic. Should have warmed up my tongue this morning before class. So what do you think, Madeline? If it's a peptidoglycan membrane, then it's what kind of a major type of critter that we've been talking about back in, what was it, previous chapter, right? This is 28, yes. See, I'm always one of those two and two. There you go. Yeah, that's fine. Prokaryotes, right? And again, I'm not expecting these words to roll off your tongue the way they do mine. So if you're worried about the spelling, at this particular point, I'm just, do you know the answer? So that's fine. Yeah, it's the prokaryotes. That would indicate to you that what was engulfed, something was engulfed, and it wasn't another eukaryote, which can happen, but it was a prokaryote. And the observation you made about the plants, yeah, so you got two really important pieces of information out of this. Very cool. I like that. Okay, that's what I mean about that whole, I always call it using the old noggin. You guys sometimes are better at this than you think. Because sometimes when you look at things, you go, oh, it just makes sense. If you just kind of recognize certain things like terminology, and I understand, that gets confusing. So speaking of confusing, we're back to the protozoans, which are all screwed up. But we're down here in the Archaeoblastida. If you're like, what the heck did he just say? I said this word right here. Archaea often means like old or ancient, like the domain Archaea. And blastida, a chloroplast is a plastid. There are other kinds of plastids, but plastids are things inside cells. I guess they'd be organelles then. Look it up. It's hard for me to remember exactly what they are, but I know a chloroplast is an example of one. All right, so archaeoplastids might be those cells that, oh gosh, I never thought about this, but maybe that word means these were the first cells that were These were organisms that arose out of that first endosymbiosis thing where cells engulfed photosynthetic bacteria. Now, I don't remember if there's an order to that or not, but I do know that in Bio 1, I believe we talk about the cell engulfing the photosynthetic bacteria first, and then the eukaryotic cell. So maybe cells became photosynthetic first, right? Think about evolutionarily speaking, that would have been easier. Everybody's photosynthetic. And then later on, you may have incorporated the aerobic bacteria and be able to respire. Or maybe what you did is you incorporated that bacteria and lost the photosynthetic cell, which is what we said probably did happen. There you go. So the reason why I'm going through that exercise with you, if hopefully you're not lost, if you are, have another drink of coffee or whatever, right? So the reason why I'm doing that is if you look in that group of organisms, remember how the protozoans are all mixed in with organisms that aren't protozoans, right? Well, there's the land plants, and I put them in red to remind you, land plants are not protozoans. They're not. Whatever protozoans are, it ain't them, okay? But notice we talked about the other day, they're more closely related to the protozoans than they are to us. So land plants are photosynthetic and they have chloroplasts, and we know chloroplasts were engulfed. So maybe that's what this is talking about, because red algae, chlorophytes, and charophytes are both the green algae. Remember that weird word algae, right? Red algae, golden algae, brown algae. Now we get the green algae. In the green algae, there are two of those protozoans, chlorophytes and charophytes. Now, chlorophytes are very much like protozoans, okay? Whatever that means to be a protozoan. Ooh, sorry. But now, charophytes are much more like plants. They're still protozoans, but remember, Aristotle, you're not right. It's not rungs of the ladder. It's spaces in between. So that's why you look at the evolutionary relationship. Let's see if my pen doesn't screw me over here, right? So let's say we look at that evolutionary relationship there versus there. We say, all right, karyophytes, which are protozoans, are more closely related to land plants than they are to other green algae. So it becomes that very odd way of how things get linked together. So I think that's what archaeoblastid means. That's cool. I think it means the first cells that engulfed something that photosynthesized. So you get this photosynthetic bacteria, along comes a eukaryotic cell, and the archaeoblastids the name would represent, these were the first cells that did that. Wonderful. Well, you better not miss any questions on the exam about archaeoblastids. Okay, archaeoblastids, we believe, are all monophyletic, so they all came. Oh, I just said what I wrote down here. Holy cow. It's always nice to come up with the same wonderful conclusion in your head twice, isn't it? Because then you know you're right. There we go. These are all from some ancestor that was the first one. It would have been this ancestor right here. All right. See, as you move on down the road, you're coming down to us. We didn't do that. So that would have mean photosynthesis was first, and then later on organisms became well, actually, photosynthesis and heterotrophic was first, and later on photosynthesis was lost. All right, so let's get off of this theoretical stuff and start talking about these protozoans. We'll talk about the land plants, obviously, in the next couple of chapters, but let's talk about the protozoans first. But let's move into the ones that aren't green. Let's talk about the red algae. Oh, these are so cool, the red algae. For one thing, they're red. And for one thing, they photosynthesize. Well, actually, the second thing. They're also unicellular. They're found in tropical oceans, coastal tropical oceans. All right, so the coastal part of that sentence, what does that tell you about where you would find a red algae? I mean, you don't tell me along the coast. I'm sorry, I should rephrase that. Tell me about the depth of the water in those areas, coastal. Yeah. I mean, we're not talking about the middle of the ocean where, you know, it's a couple of, what's the Titanic is at like 2 miles. It's just amazing to think about, right? We're talking about shallower waters. Now, relatively speaking, shallower waters can be deep. I'm not saying that shallower waters are only those things that come up to your waist. I'm talking in some cases, shallower waters can be on an order of a couple of 100 meters because the ocean can be pretty doggone deep. So it's a relative kind of term, I get you. But what I'm kind of getting at here is the whole idea behind light. All right. In order to photosynthesize, and red algae are archaeoblastids, so guess what? They can photosynthesize, even though they're red. We'll talk about that in just a second. That if you live in the water, which that's where we find these red algae, you're going to be dependent on the light getting through the water. Now, I mentioned you guys in my other class the other day that all scuba divers know that, like, it doesn't take very deep water. You can only be down 20, 30 feet. And if you cut yourself, your blood does not look red. It looks green. I've actually done that before, and it's really cut you for a second. You go, wow, that's right. It is green. And the reason why it is take a look at that chart. We say that light, this word, attenuates. Y'all know what attenuates means. Do you know what attenuates means? Cooper? Oh, Cooper's asleep, so he's online, but he doesn't know what it means. Do y'all know what it means then? I'd rather you did than the dog anyway. It's a weird word, but it explains what happens to light, different colors of light as you go deeper in the water. Fades. If you're getting ready to type the answer, I'm sorry, I stole your thunder, but fades. Attenuates. Light the fades as you go into the ocean, right? Doesn't it? Yes, of course. It gets darker. But the interesting part about it and what the chart is showing you is it's different by color. So if you'll notice over there on the right-hand side where red is, in order for my blood to look red, I'm going back to Bio 1 and photosynthesis. In order for my blood to look red, all of the different colors of wavelengths of light have to hit my blood, and the only wavelength of light that's not absorbed is red, right? It's the same reason plants are green. Okay, so if I'm down in the water, now I said I'm 20 feet, that's like 3 meters, so I'm way up here. I'm still up here where there's a lot of red light, but the red light is diminished. And it's diminished enough so that my human eyeball can't tell it. So when I'm down at just 3 meters, even though there's red light, enough of it has gone away so that I don't see the red. I see the next most prominent wavelength, and that's over here in the green side, greenish yellow. And if I remember right, it does. It has that color. And the deeper you get, it's sometimes funny when I go scuba diving. If I go down relatively deep, I'll see a fish that I recognize by a certain color pattern. I'll go, whoa. Well, that looks like that fish of the right color pattern, but everything's more muted. The colors become really kind of messed up. But this whole idea behind colors fading, and the next time you're down at depth, try that. Bring something green, like a green ball or something, and if you can get it underwater, and at the bottom of the pool or out in the ocean or something like that. It's really kind of cool to watch. I'm not suggesting you go cut yourself, but if you go scuba diving, that's why you do have your dive knife, though. You have your dive knife, so if you see sharks, you can cut your buddy, right? Got to protect. It's Darwinian, so right there you can check it out. But anyway, red algae, and even that stuff that they put around your sushi. Gosh, I love sushi. That stuff that doesn't look red. It's very, very dark in color, what we call nori. That's actually red algae. Red algae is found in very deep waters, and the reason why it can photosynthesize in really deep, I'm talking really deep waters, like way down here, at like say a couple hundred meters, well, guess what? There's no red light, right? But there's no green light either, right? All of these colors of wavelengths of light have filtered out. Now, back up to the top. Let's get away from blood for a second. Do you remember in photosynthesis, the wavelengths of light that were most important for it? Let's see how much you remember. Marie, I know I said this in Bio 1, so I'm hoping you remember. There were a couple of wavelengths that when we talked about photosynthesis, we said that these nanometers is the most sensitive to make photosynthesis work when we were looking at the photosystem. Y'all, they were in the hundreds. Y'all remember? Do you remember the colors at least? Well, that would have been the least. That would have been the least. Right? What were the ones that were the most? Yeah, the reds, right? 680 and 700, as a matter of fact. Now, if I could plot that on the chart, that's wavelengths of light up in here where I'm circling with the blue. So if you look at that, and we got a red algae growing down here at 200 meters, how does it photosynthesize? How does it photosynthesize if the light it needs, which are the reds, are gone at, gosh, certainly by 50 meters, right? Even if you throw in a little bit of the yellows they can use, you're gone there by 100 meters for sure. The only thing you're getting down at 200 meters, or even a little higher than that, depending how sensitive you are, you only are the blues, aren't you? A little bit of the greens, a lot of the blues, not even the purples. You're getting the blues. All right, so what is going on down there with red algae is they're absorbing not red light, but blue light. So since they're not absorbing red light, plants are up on land, but these photosynthesizers or plants in shallower water, they're not absorbing the red light, right? They can't. There's no red light to absorb. So they're reflecting any little tiny bit of red light. I mean, this chart is not completely accurate. There might be little bits of red light down there at 200 meters. But any bit of red light there are, what do red algae, what color of light do they reflect? What color of light do they reflect? What color of light do they mostly absorb then? So like in the plants, you know, they reflect green. They don't absorb green. They absorb red. So these guys are reflecting red. Red, right. He said twice. I'm sitting here thinking, oh, wow, she just said it twice. Yeah. So what they're doing is they're reflecting. That's why they're called red algae. Now, what are they absorbing? They're absorbing the light that's down there. They actually do the opposite of plants. They absorb the blue, right, which plants, well, algae do. Plants don't absorb, but they absorb the green. They absorb the green. They absorb the green light that plants absorb. Now, they don't do that with chlorophyll. Chlorophyll is green. Chlorophyll is green, and if you're green, you reflect green light. But they have chlorophyll. Oh, gosh, this is even more confusing. They have a molecule they don't even use. They have a helper. They have this stuff called phycoerythrin. Phyco means something to do with plants. Erythrin means red. Okay, your red blood cells have an official name. They're called, if I can spell it right, God, erythrocytes or something like that. So what they're referring to is this pigment that they have looks red, right? So chlorophyll looks green. We know things photosynthesize. This pigment looks red because it absorbs blues. If we put it into a spectrophotometer, it would absorb a lot of blue light. It would absorb a lot of green light. It would make something. You remember when we looked at photosynthesis at the spectrograph, there were two peaks. Down here in the reds that y'all figured out already, huh? And in the greens and the yellows and the blues and the reds down here. I'm going to erase this. If we did it for this phycoerythrin stuff, you would see a peak in the blue. Come on, pen. You would see a peak in the blues and the greens. Boom, it would drop off and I didn't draw that so well, but it would drop off very quickly at the red. Okay, here's what it does. Phycoerythrin is this pigment. It absorbs the blue, it absorbs the green light, it takes all that stuff and other plants don't do. Then it transfers the energy. That's the weird thing about light and plants and stuff. They're absorbing this photon thing. So they're absorbing this thing that's also energy. Oh, gosh, it's so confusing. It's all quantum. It's all quantum. It's all this weird stuff that goes on in physics called quantum. If you want to read some weird stuff, read that. Anyway, the energy that the phycoerythrin absorbs, okay, it gives it to the chlorophyll. So guess what? Red algae have chlorophyll. If you took the phycoerythrin out, they would look green. You get it? Isn't that cool? So chlorophyll is the the god or goddess, if you will, of photosynthesis. So if you photosynthesize it's like, what was that? All roads lead to Rome, huh? All roads lead to chlorophyll when you're photosynthesizing. You can be an accessory pigment like carotenoids. Why carrots look orange? Carotenoids. There was another one. There's even two chlorophylls. There's a chlorophyll A and a chlorophyll B. Xanthophylls. Some plants have an accessory pigment. These guys have phycoerythrin. Isn't that neat? So someone ever says to you, if things have to be green to photosynthesize and red algae photosynthesize, you can lay it on them, can't you? You know it. And they'll go, whoa, that's so cool. Isn't that cool? So when you're at sushi next time, order, I can't remember what kind of rolls get the nori around it, but get one of those. All right, so let's go back where the red algae, let's go to the green algae. Oh, now this is when we start really messing with the Aristotelian ladder. This is when we start putting ladders in between. So here's how it goes. Green algae are green, okay? Green algae are green. Remember, the word algae is kind of confusing. Lots of things that are algae, all right? But usually whatever they are, they're kind of simple. Sorry to you algae out there that are listening. Most of these things are found in fresh water. There are a few marine species. We have a lot of green algae here in Florida. There's a few marine species. They're not real good at dealing with the salty environment, which you would find in the marine species. You can find them in the soil. Anabina, Anabina is a prokaryote, so that's not going to work. Who am I trying to think of? Volvox fits into this. There it is. Volvox fits into this category. This weird one, I got the name about halfway down. We can try to say that. Say Chlamydomonas. See, the nice thing about science words, scientific names, they're all very phonetic. They're Latin, so you can just do the old sound it out. Chlamydomonas reinhardti. We have to put the I's at the end. That's probably someone's name, Reinhardt, who probably worked a lot in algae. You can't name something after yourself. So the dude who named this couldn't have been named Reinhardt, but if his buddy Joe Reinhardt was a good friend of mine and he died studying green algae, so I'll honor him. And then you add the I's to make it Latin, all right? So we'll get back to that in a second. So soil, they're found in, not so much in the ocean. Lichens, oh, they're so cool. Lichens are a symbiotic relationship. Lichens are in some cases, some not all, but in some cases, it's a green algae that lives in a symbiotic relationship with a fungus. Now, we'll talk more about lichens later on, but it's one of those really cool things that goes on in biology where these two critters, this green algae, a protozoan, okay, and a lichen, which is a fungus. Now, we don't necessarily know what kingdoms are going to come out of protozoan, but we do know that there is a kingdoms of protozoan and there's a kingdom fungi, and we got these two critters that are living together. They've lived together for so long that people who study lichens actually call them species. They say, oh, that you have to identify them. There's the Christmas, I think I took you out in the woods and showed you guys, the Christmas tree wreath or the Baton Rouge lichen. There's a species name for that, which totally screws up, totally messes up what we talked about species early on. And we've got two kingdoms of things that are living together. Anyway, more about lichens later. Obviously, this group is screwed up in terms of phylogeny. It's paraphyletic, right? Paraphyletic, where what we're doing is we have an ancestor, and we're talking about some but not all the descendants. Let's do a little exercise here. Let's go back and look at that again. Why is the group of Archaeoblastida, right? Why is that paraphyletic? You know, I'm getting ready to talk about the protozoan archaeoblastids. Why is that a polyphyletic phrase for me to use? Because, I mean, who am I getting ready to discuss with you guys? I just talked about one of them. I talked about the red algae. Why is that? Why am I doing this in a paraphyletic nature? Oh, that was a good way to say it. You guys wrote some good stuff in the exams, too. Some of you are very clever writers. I love that stuff. Come on, it's another question. Yeah, very cool. Yes, if it had the land plants in it, then it'd be monophyletic. But guess what? I'm not going to sit here and talk to you about land plants being protozoans. Why? Because they're not protozoans. So we're having a paraphyletic discussion today. So there, for all those people that missed the recording, you missed a live paraphyletic discussion. All right, let's go back and take a look. We've got two groups of those. Aristotle, you're not right. You got caryophytes. All right, caryophytes. Now, caryophytes led to land plants. Caryophytes are not, repeat, look at them, they're right there. They're not land plants, but man, they're close. It's so weird, though, because if you go and you say, well, in a big way, they're green algae, the next green algae they're related to, they appear to be just about as close to land plants as chlorophytes. So guess what? Take that in the nose, Aristotle. If you say, here's the protozoans and here's the plants, I can show you that something kind of there. So there. You're not so smart after all, are you? No, he was a smart guy. I would never say that to his face, even if he were alive. All right. So we'll go back to the caryophytes when we get into the land plants. All right. Let's go the other route. The coelacanths fit in that group too. I'll talk more about those later on. Let's go back a little bit. Let's go to the more protozoan-like green algae, what we call the chlorophytes. Some are unicellular, like Chlamydomonas reinhardti. There it is in the picture. Not very big, just 5 microns. But about a billion years ago, what was going about a billion years? The Earth was actually starting to warm up. Yeah. A billion years ago, what we call the snowball Earth had just ended. We talked about that a little bit when I was mentioning in the evolution of Earth. About a billion years ago, the Earth, or just before that, the Earth became a big snowball of ice and nothing was really going on. Life survived, but just barely. And about a billion years ago, it began to cool off. I'm sorry, warm up. And so when it warmed up, the ice began to melt, sea levels began to rise, started to get tropical. Plants love that kind of stuff, especially if they're coming out of the water, especially if they're coming out of the water. A billion years ago, the land plants went, I'm going to go out of water onto land for reasons that we'll talk about later, right? If you think about it, just more stuff. And these guys, the chlorophytes, especially the ones that were unicellular, said, I'm going to stay the protozoan route. And they retain characteristics of being a protozoan. Some of them did get together, right? Like Volvox. We looked at Volvox. Did we? No. Did we? No, we don't look at Volvox till plants. I actually look at Volvox in plants. That's how kind of screwed up this is, because to me, the lab manual that I've always used, typically they've always had Volvox in with the plants. So this is what becomes kind of weird about that. I leave it here because I do want to kind of get you not confused, but I want you to understand, man, evolution is this dynamic thing. All right, so Volvox can live as a hollow sphere, 500 to 60,000 cells in a cooperative living. Oh man, so Volvox has a protozoan foot. Oh gosh, not a foot. Has a foot in protozoa life. Has a foot in plant life, and is moving along the lines of cells living together, not just the colony, not just the biofilm, but cells living together, right? Living together. Now, when you live together, you know how that works. If you live with someone, if you live with a group of people, say, it's division of labor. Johnny takes out the trash on Mondays. My brother-in-law does that. His kids have a little schedule, and they earn points for all. It works, man. I tell you, it works. My kids didn't have a lot of rules, which probably explains a lot of things. So I was just like, hey, the garbage overflowing. Somebody better take that out. Anyway, and I used to bribe them. Like things they would say. They would say, Daddy. I would say, which one of you loves me more? And they would yell back and forth. And I'd say, good. Then go do the dishes. Now, let's get off of my parenting, because I certainly don't want to put this on tape. But anyway, let's go on now with the chlorophytes a little bit further. Have you ever seen a chlorophyte? I'd say raise your hand, but I can't see you. But if you've seen a chlorophyte, raise your hand. Not just Volvox. I just showed you Volvox. Have you ever seen it? Have you ever been to the beach and seen a chlorophyte? Bet you have, bet you have, bet you have. Here you go. If you've ever been to the beach, you might have saw this green stuff that floats around. Sometimes it gets on you. You go, what the heck is that? It's very thin. It's very thin. I have some in lab, but it's all dried up, so it doesn't look very pretty. So I had to show you a picture. Ulva, not the thing in the back of your throat, which is the uvula. There we go. Woo. Anyway, an ulva is the scientific genus. There's no species there for seed lettuce. That's why it kind of feels the way it the way it does. Right? You've seen it. If you've been to the beach, you might even see it. Sometimes it washes up on shore. Go walking on the beach in the morning. You've seen sea lettuce. Trust me. So the next time you look at sea lettuce and you're walking along the beach, and one of your friends or boyfriend, girlfriend, or whatever says, oh, look at that plant. It's kind of cool. You say, that's not a plant. That's a... Come on. I'd be so impressed. I was walking along with somebody and I said, look at that green, thin, seed lettuce looking plant. You'd say, well, that's not a plant. It looks like a plant, but that's actually a what? Come on, do it. Cooper's not going to answer. He's sawing logs, so he's dead asleep. You get bigger picture. It's not in the kingdom Plantae. That's a protozoan, you'd say, and they'd go, yes, who cares? Caulerpa, if you've ever done any snorkeling in the Indian, Banana Rivers, intercoastal waterway, you have probably seen something. Look what it looks like. Looks like a feather, kind of. Looks like what we'd call a symmetrical feather. That's very important when we talk about birds later on. This is not a feather, obviously, but it is a, what we call an intertidal. That's why you see it, banana rivers, and those are intertidal. Tide goes up, tide goes down. Chlorophyte. Chlorophyte. These are chlorophytes. Now, are chlorophytes plants, okay, or chlorophytes protozoans? What are chlorophytes? And you'd say they're kind of that, okay? Now, remember, I'm on the protozoan side of green algae. The caryophytes led to plants, and I'm not talking about that anymore. We're going to talk about that when we get into the plants. I'm going to go back and pick up and go on with the plants. Anyway, we're going along the green algae side. These are protozoans. We don't know what necessarily. They might be kingdom archaeoblasted up. They all finally figured out. They may have. I don't get the protozoan weekly. But the point is, these organisms here, what have they done as far as the chlorophytes? Because even though the chlorophytes don't lead to the land plants, does not mean that they don't lead to some interesting innovations in evolution. For example, the formation of colonies, which we already talked about. Volvox has done that. Multicellularity in ulva, not tissue. Ooh, becomes Aristotle. You knew nothing about. Oh, well, he did. I'm sorry. Back to Aristotle. Oh, why do I apologize to a dead guy? Anyway, there are a lot of ladders, a lot of steps in between the ladders. Multicellular, not necessarily tissues, but now Callirpa does something really kind of bizarre. Really kind of bizarre, because we're getting the fungus. Fungus, oh gosh, they're the weirdest critters on the planet. Anyway, what Callirpa does is multinucleation. Now, it sounds like a big word, but look what happens. Here's what happens. I put it in the arrow. You get the division of the nucleus without the cell. So that leads to multinucleation, doesn't it? It leads to a cell. I'm not going to draw a cell with a bunch of nuclei. You know what I'm talking about, right? Supercells. Now, what I want you to answer for me, though, is why is a multinucleated cell, why could we call it a super? Why could we call it a super cell? What makes it super? Not super in the terms of numbers. Don't tell me, because it's got a lot of nuclei. Yes, I'm talking about super in terms of evolution. These are evolutionary innovations. Right here on the base, man. I tell you, there's all these kinds of things I'm talking about are going to build up when we go to the different critters. That's why this whole class ends in this crescendo of diversity. So what's the benefit if you have a lot of nuclei in a cell, one cell? Why are you a super cell? What's in nuclei? How does that fit into evolution? How if I have a lot of it? A, B, C, D. Come on, guys. We all know we have one nucleus in one of our cells, so it just means that everybody's got to be a supercell. But some critters came up with an interesting innovation. They came up with supercells. I really want you to kind of think about this, because this is going to become important when we get into fungus. Now, Callirpa, not fungus. Not fungus. This would be some example of convergent evolution. But if you think about a cell that has many nuclei, and each of those nuclei have genetic information. Now, let's say each of those nuclei were created through meiosis. We all know meiosis creates copies of cells, yes, but they're not identical. Meiosis is the key to evolution, is it not? Right? Variability. Gotta have variability. So guess what we have in supercells? We have lots of genetic variability. We have, oh boy, I'm stretching it. We would have a population of nuclei, wouldn't we, in a cell? Now, do you think that cell would be super in terms of fit? Do you think that cell with all that genetic information, nobody says it's all good, all bad. Nobody say anything about, quantity, but let's just say there's enough that fits the category being fit. Do you think that cell with a lot of genetic information might be more fit than, say, another cell with less genetic information? What does a lot of genetic information, what does a lot of variability allow you to do? What does it allow a population to do? Flip the question around. If you don't have variability in a population, then what could potentially happened to you? Wake up. Come on. I know you guys know this. Don't just if maybe there's a lag. Cooper's snoring, so he's not going to be able to help you. It's the same thing we talked about, right? I'm just going to assume I'm not getting an answer because either I'm not, this is not going off into cyberspace and nobody's getting it, or you guys are just being shy. But if I'm a supercell, don't I have a lot of variability in all those nuclei? I'm one cell. I am the unit of life, right? We don't get units of life until we get to the cell. That's bio 1. Did that rhyme? I should write that down in a song or something. Anyway, if I've got a lot of genetic variation in me as a cell, all right, give me some icon that tells me you guys were coming up, that was formulating. Even you don't have to, but give me some sort of a, it reminds me. Anyway, give me some sort of an icon or some indication that lets me know you guys were kind of going along those lines, because this is so important in fungi. If I'm a cell with lots of nuclei, don't I have lots of genetic information? Haven't I just taken the concept of a population? No? Haven't I just taken the concept of a population of wildebeests? Yeah? Which are all different. Okay, and I take that concentration of wildebeest, and what I've done is I've said in a supercell, what a supercell does is the same thing, only it does it with lots of nuclei. One cell with many different nuclei. Each nuclei has different genetic information. It's kind of like wildebeest at a microscopic level, right? That would allow for that cell to survive, because it has lots of genetic information to draw upon, things that it can use to be immune to disease, things that it could use to grow faster than the next cell, things that it could use to more efficiently use the nutrients in the soil. Different, remember with DNA, it's information on how to survive in the environment. The more information you have, the better, right? Madeline? The more variation you have, the better. Yeah. So guess what goes on in supercells? That's what they've done. It's fascinating. Fungi do this, man. They have cells with many nuclei that some are sometimes fused together like ours do. Now, speaking of us for a second, how come we're not supercells? Does that mean we're not fit? I mean, supercell seems pretty cool. I'd like to have supercells. But, I mean, does that mean since we don't have supercells, because we do only have one nucleus in our cells, does that mean that these guys are more fit than us? Callirpa? I think I could probably mush a callirpa. Oh, the holy cow, I could eat one of those. But does that mean that we're not fit? You're thinking, doggone it, he's making me go back and tie in the concepts from the early part. There you go. I love that. Yes. It's all about environmental reproduction. Yes, you are. Yes. Oh, I love this. Okay. So that, when you look at stuff like that and you go, wow, supercells, that seems like it'd be a pretty cool idea. These guys have done pretty well. Callirpa's been around a while. They survive. Fungi, don't get me into fungi, and they're multinucleation. We know that they do pretty doggone well. The biggest critter on the planet is a fungus. More on that later, okay? We'll talk about that. It's hard to believe. People say, what's the biggest organism on the planet? Tell them it's a fungus. All right, so yes, it's the environment and the way they reproduce. So evolution does not create perfect critters. It creates organisms that are good enough. All right, done with all of them. Now we're in the Uniconta. I think it was Logan said something the other day about conta meaning pole or flagella, okay? If you're uni... my wife's cousin, he's a cool guy, Junior, but he is, oh my goodness, he is unibrow. He does get it tweezed, thank goodness, but it's amazing. Anyway, unibrow, how many eyebrows you got? So anyway, Uniconta, if conta is flagella, or flagella, pole, right. How many flagella you got if you're a Uniconta? There we go. You and I, we are uniconta. Name the cell in my body. Probably not in, well, no, not, okay, not in y'all's body. Anyway, just get digging a deeper hole. The cell in my body that has a flagella, because I'm a unicont. What cell in my body has a flagella? Yeah, there we go. There we go. All right, good deal. Now, while we're sticking on that for a second, break on down. I know I didn't, I'm going out of order a little bit. How about opisthoconch? You know what cont is, right? Right? Opistho or opith means behind, not in front. No kidding. So somebody who can, if you can't, tell me, draw on the screen a cell with the flagella behind that I would recognize as Madeline telling me what is the cell in my body that has flagella. I want you to put the flagella in the right place on sperm. If you're going, what the hell is he talking about? Oh, now it tells me something about video quality getting messed up. So I'm going to assume everybody's seen sperm. The flagella in the back, isn't it? Okay, now you don't have to have a flagella to be in the uniconts. You have to have a projection, a pole. So it could be a pseudopod. Look up there, amoebas. Remember I told you the word amoeba is not monophyletic? Does that make sense now? We talked about amoebas up in the radiolarians, the rhizarians. There we go. Because we said they had pseudopods, thin. This is definitely not thin, but blunt or big pseudopods. So what? Doesn't mean you're related. Because here we are now in the uniconta, and specifically, we're going to go into something called the amoebozoans. Where am I in time? Okay, good deal. All right, so we got, now look at this whole paraphyletic thing again. In this group of uniconta, look who we got. Protozoans. No kidding, that's the chapter in. That's the guys in yellow. We'll talk about the guys with the black box around first. Fungi. Those are not protozoans. Really? Animals, us, you, me, blah, blah, blah. Look, we're more closely related to choanoflagellates than we are to something that's not a protozoan. We're more closely related to choanoflagellates than fungi, and certainly than land plants, all right? Enough of that? Cool, there's some really bizarre things in this group, right? And that's going to these guys first. These are obvious. Amoebas have pseudopods, but they're not thin, they're big, they're thick. It has a contractile vacuole. Now, in the lightning round, tell me what type of environment. Just give me the first 3 or 4 letters. You would find an amoeba because it has a contractile vacuole. That's what CV is. It doesn't have a curriculum vitae. Yes, what is true, but I'm talking about tonicity. I'm talking about solutes on either side of a membrane. Chapter, what, 5, I think? Three or 4 letters. One of the roots, one of the beginnings has 3. The other 2 have 4, and they almost sound alike. Come on now. What do contractile vacuoles contract out? What do they get rid of? What do they pump out of the cell? They pump water. So that must mean what? See, this is where in class I could look at you. That's where you got, maybe y'all put your cameras on so I can look at you. You know. So if they're pumping water out, what's that mean about the environment that the amoebas live in? Come on, guys. Time's ticking. You got to move on to slime molds, which are really bizarre. Which way's the water going? Out of the cell or into the cell? Wow, thank you. Yes, so contractile vacuoles mean organisms live in a hypo. Thank you, Madeline. Yes, thank you, Marie. Yes. No thanks to Cooper. All right, he is woken up, but he's not going to answer. So slime molds, look at these doggone things. These are slime molds. We call them molds because they look like molds. But guess what? We're not in fungi, are we? We're still in protozoans. These things, even the name is confusing. Mycetozoans, if you're a mycologist, you study fungus. Zoan, you go to a zoo, you see animals, fungus animals, mycetozoans. They move. That's where we get the name, the zoan part. Things that move, not all animals do, I get it. Things that move are normally associated with animals. Why? Because animals move lots of different ways, at least macroscopically. Right? We can fly, we can walk, we can jump, blah, blah, blah. So anyway, things that move are, we think they're animal-like. But they make spores, like fungus. Oh, there you go. Once again, in the rungs of the ladder. Oh, is it the rungs of the ladder? Doesn't have to be the rungs of the ladder. Sometimes things look alike or act alike, whether they look like this. Doesn't that look like a Cadbury egg in the upper left? Don't look, if you unwrap that, there'd be some chocolate cool thing in there. Can you imagine me in the woods and thinking, ooh, Cadbury egg? The other ones, but anyway, just because something looks like something else does not mean it's related. True, false. True, false. Just because you look like something else, you must be related. True, false. Just put TRF. Of course it's false, right? Of course it's false. Yeah. Doesn't mean you're related. Porpoises and sharks. Boom. Let's move on, right? These things look like molds. They look like fungus. They move like animals. But guess what? They ain't. They're protozoans. Now, why is he harping on this? Organisms are adapting. These critters are being animal-like. These critters are making spores. So other ones are doing that. And guess what comes out of that? Things like animals, things like fungi. All right, now, these guys, ooh, you look on YouTube, type in Plasmodium slime molds or type in weird stuff in the sewers of New York City. You will see tons of pictures of slime molds, okay? Slime molds defy, there we go. Try that word, Steve. Slime molds defy what we think things should look like, I think. I think that's why we're so fascinated with them. That's a slime mold in the upper left. That blue thing is a slime mold. Those red lollipop-looking things. No, those things, oh, remember you can buy them at Starbucks. What they call cake pops. Those things that look like, those are not cake pops anyway. Those are all what we call plasmodial slime mold. Oh, are you kidding me? There's two roots of confusion here. They're not molds. We're still in protozoans. Maybe they're mold-like, right? Could be relationship, could be convergent evolution. You have to look at that. And this plasmodial word, because plasmodial is the phrase we use to refer to what the orange thing is doing. It's streaming along as something we call plasmodium. Careful with that word. In lab, we looked at a plasmodium. Plasmodium falciparum. It causes a disease, right? Plasmodium falciparum is another protozoan. You saw it in red blood cells. It's a blood disease. What disease does Plasmodium falciparum cause? For $1,000. I should get the Jeopardy thing. Dun, 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 dun. We need to finish fungi today. I hope not. I'll talk about it. Look it up on your. Be on the web past your net. Come on. Plasmodium falciparum. Where are we today? I'm supposed to do 29. Yeah, no, that's 28. Oh, good. Come on. No, not STD. Give myself. No, you couldn't get it through STD. I guess you could. You shit blood. But anyway, who's the normal? Plasmodium falciparum, which is spread by a mosquito, a vector, causes what disease? It comes from an old Latin word that means bad air. Yeah, there you go. That's how they figured. Yeah, that's how they figured why people were getting sick. That's where the word comes from. Bad air, malaria. All right, now don't confuse those. Why are the words the same? I don't know. Scientists just like to do things like that sometimes. Anyway, what is a plasmodium? It's non-walled. So this is one big giant cell, multinucleate. Multinucleate. We already talked about why that's good. It's a mass of cytoplasm. Now, what these things have done is they undergo this process of synchronous mitosis. Everybody at the same time, boom, boom, makes nuclei. They don't do cytokinesis, right? Mitosis has got two parts. Karyokinesis divide the nucleus, then you divide the cell. They just don't do the second part, okay? And this is their feeding phase. They move out throughout the world. That's why they're in the sewer things. Right? This thing is moving down the sewer walls. What it's doing, it's moving along and ingesting bacteria and other organic material. Other organic material could be anything. Could be feces of something else, right? Anything that's organic Could be food. So these guys are moving along doing that. You don't have to worry about it unless you're one of those. Right. So they look cool. They look bizarre. They look like the world's being invaded, but they are not. Now, what's really another thing that's really cool about them is they're not immune to the environment like anything else. Even living in the sewers in New York, there are different kinds of environments. Sometimes food, moisture. Sometimes important things like that are scarce. So during this time, guess what these organisms do? Like a lot of organisms that we talked about, what was it, anthrax? Who was it the other day? Oh, I said cysts, didn't I? There, golden algae forms cysts when the environment turns bad. I already said anthrax will form spores, which is a cyst essentially when environment turns bad. When food or moisture is scarce, guess what these guys do? Same thing. They form sporangia. Sporangia are just things that make spores. That's why that's not a cake pop. Those little red things are spores. Guess what's inside the blue things? Guess what's inside the blue things? Whee! Spores. How many? Hey, take a guess. Lots of spores, few spores. I know it's a dumb question. Just humor me. Lots of spores. You can up arrow, be or whatever, or few spores. I mean, heck, you look at the cake pop thing, and I think the answer is obvious, isn't it? I would guess. All right, let's take a look at how they reproduce. Why? Because it's just so bizarre. Anyway, start up at the top in the upper left with the zygote. We start there because that's something we're very familiar with, right? Because the rest of this is a little weird. And a zygote is what a zygote is. A zygote is a diploid cell. That's the definition. Now, you start off as a zygote like everything else. Once that cell is fertilized, once those nuclei fuse, guess what? You got yourself a zygote. All right, so we're diploid. That's why we're in orange in this picture. That's what the orange means. So what happens is synchronous mitosis without cytokinesis. We make this big, look at it. Look at the picture there. I mean, would you ever think that's an animal? Or excuse me, protozoan. Not anywhere near an animal. Well, yeah, kind of near an animal. Isn't that weird? Anyway, so this thing is moving out, and the environment gets bad, so it starts to basically bubble up during these harsh conditions. These young sporangium form, and they form a stalk, and inside of the sporangium, they make spores. And guess what you do with spores? You make lots of them, but you don't just make lots of them. You make lots of different ones. Never forget that, folks. Meiosis, variability. Mitosis, numbers. Mitosis, numbers. This is mitosis, folks, technically. You don't have to have cytokinesis to get mitosis. So that's mitosis. So mitosis. All right, that's why that organism is a little more than diploid, but whatever. In the process of meiosis, you produce spores. Number one, they're resistant. They're harsh. They want to make the environment's crappy, okay? But they are haploid. They are haploid. So they're the don't screw this up. They're not the gametes, but they are the gametish adjacent cell. All right. Anyway, they find a condition that's right. Maybe now, maybe later, maybe never, and they germinate. Now, when they germinate, what comes out of those is amoeboid and flagellated cells, which can switch between each other. Now, these aren't sperm and egg. These aren't male and female. There is no male and female among creepy crawly things in the sewers of New York City. But what you have is this interesting process by which cells can switch back and forth. And they're going to do that based on conditions. Perhaps environmental conditions favor more flagellated cells, right? And perhaps environmental conditions favor, at the same time, fewer ambioid cells. So they switch. Beautiful. The environment you're in is going to dictate why you do these weird things. Anyway, they do fuse, and we're back to a zygote. Isn't that a weird thing? Okay, those are not the weirdest of the, well, they are weird. These are weird too. These are what we call cellular slime molds. How close am I to the end? Oh, I think I got time. Cool. Cellular slime molds. Again, they are not molds. Very simple developmental system. Why is that so important? Because guess what? So do we. Well, we don't have a simple one. We have a developmental system. So if we're going to study something complex, if you're studying math, you didn't start with calculus, did you? I bet there's no way you did. I mean, if you're like me, you started way, way, way back with arithmetic. That's what students say. So, no, I'm not really good at math. I'm like, sometimes y'all aren't good at arithmetic either. Adding, subtracting, multiplying, and dividing. But anyway, if we study a simple developmental system, then we can kind of use that to make assumptions or make predictions about bigger, more multicellular, more differentiated systems like ourselves. That's why sometimes when you see do you know that the federal government is spending money to study mold? And you think on the surface, well, that's kind of stupid. Mold is mold. But then you get into it and you realize, you know what? For one thing, in this case, they're not studying a mold, regardless of the taxonomy of that. But what they are studying is something that could tell us something about ourselves, right? Because, I mean, again, arithmetic tells us something about calculus. The numbers still, anyway, are supposed to add, well, they do. They do. 1 and 1 is 2 in calculus, whether, and in arithmetic, is it not? Now, what's cool about these critters, follow the numbers 1 through 6, so we're in the upper right. Individual organisms behave as amoebas, so that's an amoeba, moving around, ingesting stuff with its pseudopods, just like we saw in the previous picture. Now, when things get rough, and this is kind of misleading too, because sometimes things get rough, sometimes things get good. You have to think about forming spores as a way of dispersing, but also can be used as a way of surviving until, conditions are right. So everything's cool. It moves as an amoeba. But when things get scarce, individual amoebas come together. Okay, so what? So we have a colony. That's what's happening in 2 and 3. That's all those weird. These are all microscopic. That's a quarter of a micron, or a quarter of a millimeter. Excuse me, 250 microns. So this is happening in half of the space when you're looking at the 40x of the microscope. All right, now during these times when the individuals are aggregating, they form this slug-like thing. And then this slug-like thing, 4, 5, and 6, begins to stand up. And then at the end of it, it forms a sporangium. All right, now a sporangium is a fruiting body. It does not make fruit. We're referring to fruit here as reproduction. So inside that bulb is going to be a bunch of those spores, just like we saw in that picture, okay? And now the cool thing about this as far as cell differentiation and multicellularity, which is why we study these, is when they come together, they decide, quote unquote, somehow it's figured out that you're going to be the stalk, you're going to form this part, you're going to form this part, you're going to form the inside that forms the spores. In other words, they divide up jobs, and we're really sort of interested in how they do that, right? Because isn't that what our cells do when they act as tissues? Here's the reproduction of them. All right, so where's zygote? Here we go. Zygote grows by consuming haploid amoebas. Hold on, we'll get to that in just a second. Anyway, zygote undergoes meiosis, variability. We make amoebas. Amoebas move around, but when things are good, amoebas move in this big circle to the left. So asexual reproduction. Remember, asexual is no variation. But think about that. The environment's not changing. At least it's not. Your environment's not changing. If you don't have to worry about things that could cause detriment to species because you pretty much figured it out, then go for the gusto, right? Go asexual. Boom, 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 start populating. The whole idea is that you don't want anybody else to have the resources. You want to take over the environment there. It's all very much dog-eat-dog world, even if you're down to protozoan level. So asexual is cool if nothing's changing because you're all the same. But all you're really worried about is quantity. You're just trying to take over. Now things are changing. Don't know what's going to happen. Can't predict that. We talked about that in evolution. But during this time when things aren't changing, you're making spores that are asexual. So they're genetically the same. Right? Boom, 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 and they're just germinating and just going around and invading the subways. This is a one down at the bottom. This is it's like a little weird mouse. Diclostellium discoideum, a migrating aggregate of cells. That's how these things move through the environment. All right, once again, division of labor during this asexual reproduction, though. Some of the cells form the stalk, others make the spores. How do they figure that out? We definitely want to look at that. During sexual reproduction, those amoebas that eventually aggregate together when food is low, during sexual reproduction, what those amoebas will do is some of them, not all of them, some of them actually get eaten by the zygote, and that's where we started at. But those amoebas there are actually going to fuse, and that's going to be the fertilization event, when two amoebas fuse together. So solitary amoebas in the feeding stage, and then they're going to jump into this meiosis when food gets low. All right? Actually, wait, hold on just a minute. No, they say the aggregated amoebas when food, so they do asexual when food is low. Ooh, so they use the numbers game. Don't worry about that. Just anytime when the environments change, organisms are going to respond to it by either increasing reproduction or decreasing reproduction. All right, here's the typical amoebas. The tubulinids is the ones, Amoeba proteus, you've seen under a microscope. This is a very diverse group. Found all over the place. Active predators. These pseudopods not only good for moving around, yeah, but you can also engulf things with them. I have a cool video on Blackboard. You can see an amoeba engulfing something. Some are detritivores. Detritus, if you went into a lake, even some lakes on campus, and you dug down in the shallower water, you'd find muck, right? Decaying leaves and that kind of stuff. That's detritus. They're critters. That's still usable organic material. They're critters, maybe not horribly palatable. Heck, vultures aren't horribly palatable. Do you know vultures defend themselves when they're aggravated? One of the things that they defer predation or being messed with is to throw up on you. Can you imagine what throw up of a vulture must be like? They're already eating roadkill. All right, so that's kind of what detritivores do. They eat that sort of stuff. Some amoebas are problematic, aren't they? Right? Here's two of them. Entamoebas, amoebas that occur inside other organisms. So yes, parasites. And in some case, vertebrate or even invertebrate parasites. The one in the purple, Naegleri fowleri, is the brain-eating amoeba. The purple, which one's the amoeba? Well, anyway, I can't remember. Is the reddish the amoeba? Regardless, the open space is the part of the brain material that's been eaten by the amoeba. Having part of your brain eaten is not a good idea, obviously. 97% fatal. This is the amoeba that lives in as a cyst down in the bottom of the waterways, Indian River, down in the muck that's down here. There are cysts. Okay, now when you're kicking your feet in water, you stir up the sediment. When they stir up the sediment, those cysts germinate, so they become active amoebas. They get up your nose many times by jumping in the water, so when you jump in feet first, they get shoved up your nose as amoebas, and they actually crawl along your olfactory nerve, the nerve that goes from your nose to your brain, and then they get to your brain and start doing that. 97% fatal, usually found in warm, stagnant water. So it's going to be in the summertime in stagnant water, water that's not moving. One of the girls that got it a few years ago got it in the Indian Banana River, and we were puzzled initially because the river flows, but her friends were swimming in some area off to the side, which had become stagnant. About a half dozen species. No, it's bubbling, right? There's not much sediment going to collect in the spring area as much. As long as the water is cooler, you know, this would be most problematic in the summertime. In some of those areas you might not want to jump into anyway because, gosh, the water is probably just as warm as the air. You felt those, right? Those areas you jump in and you go, man, the water's no cooler. I remember in the Philippines it was that way. The rainwater was just as hot as the air was. The one down below, Entamoeba histolytica. So basically it lyses cells, causes amoebic dysentery. Amoebic dysentery is a gut disease basically, but the problem with amoebic dysentery is diarrhea once again. So many times it's the symptoms that are deadly, right? Diarrhea is a, you're losing water, dehydrated. Any questions on those guys? Not everybody's nice to us, right? The opisthokonts with the flagellum in the rear, that's going to include the fungus, the choanoflagellates, and the animals. I'll talk about the choanoflagellates in the animals. Yes, I know it's a protozoan, but I like the connection there better. All right, so what I want to do now is just last two slides is just kind of talk about protozoans and their place on Earth, okay? Some protozoans are symbionts. In the picture on the right is a picture of coral, and coral is an animal. And in the tissue of coral, there are protozoans, dinoflagellates, I believe, which some of the protozoans we talked about, that are called zooantheli. Okay, the green cells in that picture are going to be the coral cells. The red are the zooantheli, and that's a protozoan. They live in a symbiotic relationship because what they do is photosynthesize. So, you know, animals don't photosynthesize, but the green coral cells gain the benefit of the red ones photosynthesizing. They gain food. And the zooantheli gain a benefit of a place to live, and the coral provide them with necessary materials to, you know, so they can sustain life. Interesting. That's why corals look the color they do. right? We'll talk more about corals when we get into the animals, all right? Wood digestion, for example, the one down below, the hypermastigote is a protozoan that's found in the gut of termites. That's why they can eat your house. They can't digest the cellulose, but these guys can. Some are really bad, like Phytophthora. That's the tough one because there's a pH in the middle. Phytophthora infestans causes the potato blight, which caused most of the potatoes in Ireland to be destroyed, and that's what led to lots of the Irish migration. Potato blight occurred in the 1860s, I believe. I know we're at time, but I just have just about one more slide to go, so just stick with me if you would. If you need to go, I understand. About 30% of the world's aquatic productivity comes from protozoans. Okay, very quick lesson on ecology. Down below is what we call a food chain. Very rarely is it food web. Very rarely is it a chain. Down there in the lower right, every ecosystem food web must have producers, photosynthesizers. They must have organisms that can take sunlight energy and turn it into food, because unless you can do that, you're heterotroph, right? So what we mean by 30% of the world's aquatic productivity, a third of the productivity that occurs in the ocean occurs from protozoan producers. Protozoan producers are photosynthesizing. On land, it's different though, right? On land, we got a lot more green plants. That's because protozoan producers tend to be planktonic. They float, right? So they provide food for the herbivorous plankton, who provide food for the carnivorous plankton. Then, of course, the prokaryotes are producers, or some of those that photosynthesize. But a third of the world's oceans comes from protozoan producers. And protozoans are very susceptible, in some cases, to environmental perturbation, to changes in temperature, pH, chemicals. So we've got to be careful, right? A third of the world's aquatic productivity. So all that leads up to the top consumers, the whales and the birds and all that, right? So here's the problem we're at. Sea surface temperature, global climate change. It's not global warming. That's part of it. How much of that is human caused? Well, if we look at the data, regardless of how much we argue that it's human caused, and it is, folks, 97% of climate scientists say that anthropogenic factors, it's supposed to be IC at the end, cause human climate change. That means humans. Humans are responsible for the dramatic nature of climate change. Yes, the Earth's climate has changed. It changes through time, but we're the ones pushing it. All right, the problem that we're seeing here is in areas in which the surface temperature has changed considerably, other than South India, which I figure out why that is. But anyway, if you look at like the North Pacific, NP, the sea temperature has dropped. See, global warming, it's not right. It's global climate change. Sorry, my mower guy's outside. But notice that in the North Pacific, the chlorophyll changed. It's dropped. Notice in most of the world's ocean, the sea surface temperatures have changed, either warmer or cooler. Again, disputing that global warming, global climate change. But other than the North Indian and the South Indian Ocean, that's probably because that's a relatively small ocean, I'm thinking. A lot of nutrients being dumped in there. The chlorophyll change has dropped. That means that the protozoan producer population has dropped. Less protozoan producers, less food in the ocean, less for things to eat, less for us to eat, right? All right, I'm sorry, I ate up 3 minutes of your time, but that's it for protozoans, okay? Marie, do you have any questions for me? Cooper's out barking at the guy mowing the yard, so he doesn't have any questions. No?